Hi there, welcome to Medical Microbiology. So today is our first lecture. I will explain what materials and topics will be covered in our class. In this class, you will learn about microorganisms, especially the one that can cause disease to humans. You will learn the structure, the characteristics, and classifications of microorganisms. You will also learn the infectious disease, the cause, the effects, and also the distributions of the diseases. You will also learn to culture and identify bacteria and also diagnosing infectious disease. You will also learn how to prevent disease, how to use medications, and how to cure the disease. And finally, you will also learn how human body defense mechanisms fight against the disease and also create immunity. The microorganisms that will be studied in this class will be viruses, bacteria, protozoa, algae, fungi, and helminth, parasitic worms. These microorganisms, they have roles in the environment. Most of the roles are actually beneficial for environment, for humans. The first beneficial role is many of the microorganisms are actually producers. For example, algae is actually the primary producers of oxygen. In average of 55% of oxygen in this environment is actually produced by algae. The second beneficial role is many microorganisms are decomposers. Bacteria and fungi are the primary decomposer in our environment. They decompose dead materials into nutrients that will be beneficial for other organisms like plants. The third beneficial role is many microorganisms can be used in biotechnology. For example, many bacteria and yeast are used for making food like cheese, yogurt, alcohols. They can also use for making drugs or medicine like antibiotics and also to produce vaccine. The fourth beneficial role is many microorganisms, especially bacteria, are used for creating or altering the gene of other organisms. And the organism that are altered is called a GMO, genetically modified organism. They can also be used to create a new cells, the cell that alter their gene to produce enzymes or hormones like insulin. The fifth beneficial roles of microorganisms are They can be used for cleaning environment. This is what we call the remediations or bioremediations. So some bacteria is actually able to clean environment. Okay. And the last role, which is actually disadvantage of these microorganisms, is that some of them are causing disease. The disease that caused by microorganism is called the infectious disease. The infectious disease are caused by microorganisms. There are about 2,000 microbes that can cause infectious diseases. However, from only about 2,000 microbes, there are in average 10 billion of new infections per year worldwide. And from these 10 billion new infections, there are about 12 to 13 million of deaths per year. If we look at human body system, the respiratory system is actually the most sensitive system that easily attacked by the infectious diseases. Okay, so about 26% of infections is actually respiratory infections like pneumonia, influenza. About 18%, okay, is AIDS, which is in the blood. And then about 17.5% is infections of GI tract, like diarrhea. TB accounted for 11% of infections in human body. And then malaria, 9%, measles, about 7%. Hepatitis B is about 5%. So these are the body system that usually affected by the pathogenic microorganism. I think it is enough for the first lecture. Thank you. We will meet again on the next lecture. Goodbye. Hi there. Welcome to Medical Microbiology lecture. In today's lecture, we will learn about scientific methods. Scientific method is really important when we learn microorganisms and infectious diseases. What is scientific method? Scientific method is a general approach to explain a natural phenomenon. The scientific methods is usually start with questions, wonder, observations. And then from that, questions, wonder, observations, we formulate hypothesis. What is hypothesis? Hypothesis is a tentative explanation that can be supported or refuted by experimentation or tests. After that, we perform the experiment to test the hypothesis. When we perform the experiments, we will produce data and we will analyze the data. After that, we'll produce the report, the conclusion, and usually the scientific community will meet to discuss the results. And then after several meetings, conferences, they can produce what we call the theory. Okay, when the theory is become accepted, then it's increased the level into what we call the principles or law. So principles or law are the highest level of scientific methods. This is the illustrations of cycles in scientific methods. Again, start with the observations, wonder, questions, and then we develop the hypothesis, and usually this hypothesis is related to what the research area. And then after that, we perform the experimentation to test the hypothesis. We produce the data, analyze the data, produce the report, conclusions, and then, start again. So we still need more information, then there will be more question, wonder, and then more research, more hypothesis, experimentation, and etc. until it's produced the theory. And finally, it can also become principle, okay, or law. So this is the cycle in the scientific method. This is an example in microbiology. So we know in the history that the early belief, people believe that life arises from non-living. Okay, so from non-living will become life. And this theory is called as the spontaneous generation theory. So many people believe that. And then some scientists do not really believe that because based on their observations, based on their wonder, they know that life is actually coming from previous life. Therefore, okay, they produce, the theory is called the Biogenesis Theory. And the scientist that produced the theory is Louis Pasteur. Okay. And this biogenesis theory means that life comes from previous life form. And this theory actually refutes the theory of spontaneous generation. Louis Pasteur also proposed a theory is called the germ theory. The germ theory is the theory that says infectious diseases are caused by germs or microorganisms. But on that time, Louis Pasteur did not have the ability to perform the experiment or to prove his theory because during his life there was no methodology that able to culture the microorganism, and there was no microscope yet during his time. So he unable to prove it. But the next generation of scientists will prove his theory. Thank you. So we will meet again on the next lecture. Goodbye.
Hi there. Welcome to Medical Microbiology. Today, we will learn about some historical discoveries in microbiology. These discoveries include biogenesis theory, the germ theory, microscopy, scientific method, microbiological technique, vaccination, nosocomial disease, septic technique, and sterilization. We start with the biogenesis theory and the germ theory. Both theories were formulated by Louis Pasteur. The biogenesis theory disproved the doctrine of spontaneous generation. Now, what is biogenesis theory? This is the theory that states that life is coming from previous life. And this theory disproved the spontaneous generation that mentioned life is coming from non-living. The second theory, the germ theory, states that infection is caused by germ or microorganism. However, Louis Pasteur at that time unable to prove the germ theory because he did not have the equipment to prove his theory. The next discovery is discovery of microscopes. The first microscope was developed by Anthony van Leeuwenhoek. He made a single lens microscope to observe microbes. So basically, Anthony van Leeuwenhoek was the first person that observed the microbes. He was also known as the father of microbiology. The development of microscope by Anthony van Leeuwenhoek marked the era of microbiology. So after his creation, then microbiology have been developed so fast. The next discovery is microbial technique. This technique was developed by Robert Koch. He was the one that also culture bacteria. He was the first scientist who make a pure culture of bacteria. Okay. And this scientist was the one that proved the germ theory. So he was able to prove that infections is caused by microbes or germs. The technique that he used to support the germ theory is called the Koch postulate. Now, what is Koch postulate? Koch postulate is a series of experimental steps that verified the germ theory. What is the first step? So the first step is he take the germ from the patient. So all the patients that have the same disease will have the same pathogen. So therefore, he take the sample from each patient. After that, he isolate the pathogens into the pure culture in the lab. So he developed the pure culture of this pathogen in the lab. The next step, that pathogen will be reproduced in the lab, and then he put the pathogens or the germ into test animal, or experimental host, usually using animal. The next step, then he isolate the pathogens from the test animal, and then he identify that the same pathogens is actually found in that host animal. And these pathogens will be the same as the pathogen that present in patient, in the patient. So this postulate or this experimental step prove that the infection is caused by germ or microorganism. The next discovery is vaccination. Edward Jenner was the pioneer of the vaccinations. He created a vaccine, which is a smallpox vaccine, and this vaccine became the first world vaccine. This first vaccine was developed from cowpox. Okay, so cowpox is a type of virus that infect cow. The name of the virus is variola vaccinae. That's the reason why the name of vaccine is actually coming from the name of this virus that infect cocks. It's a cow. It's called the cowpox. As we know that now the smallpox is considered as the disease that has been eradicated in this world by vaccination effort. So vaccination works. The next discovery is discovery of nosocomial diseases. Okay, so what is nosocomial disease? This is the disease that people get when they staying in the hospital. So basically, hospital is the place where some people get a new disease from other patient. So this discovery was started by observations, okay, done by Oliver Wendell. He observed that mothers of home birth has fewer infection compared to mother that give birth in the hospital. Okay, so mother that give birth in hospital, they actually carry disease from the hospital. Right? So they get disease when they are staying in the hospital. And then Dr. Ignaz Semmelweis correlate this disease, with the infection that actually carried by the physicians that coming from autopsy room to the maternity ward. So Ignaz Semmelweis prove or observe that the doctor actually transfer the disease from one patient to another patient. So this is what we call the nosocomial disease. From that observations, then coming the next discovery, which is discovery of aseptic technique. This is the technique that was developed by Joseph Lister. So he introduced the technique to prevent infections in hospital. So this is in hospital setting. So basically, Joseph Lister introduced the technique for doctors and nurses when they dealing with patient. So this technique is called the aseptic technique. The next discovery is discovery of sterilization. This was started by observation of John Tyndall and Ferdinand Kahn. Okay, they found that there were a type of microbes that actually very resistant to control, to cleaning, for example. So they still strive, they still grow, even though the place maybe has been cleaned with some disinfectants. Okay? So they call this structure that is very resistant to the controlling agents or chemical that used for cleaning is called the endospores. So endospores is the highest resistant type of microbes. This is actually part of bacteria structure. By finding this is actually give the idea to other scientists to develop what we call the sterilization technique. Sterilization technique is the technique that will remove all type of microbes, including the highest resistant one, which is the endospore. Example of sterilization is using autoclave. So autoclave used for sterilization to kill or destroy any type of microbes, including the highest resistant, which is the endospore. I think that's enough for discoveries in microbiology. We will meet again on the next lecture. Goodbye. Hi there. Welcome to medical microbiology. Today, we will learn about taxonomy and classification. What is taxonomy? Taxonomy is the formal system used for organizing, classifying, and naming organisms. The system was originated by Carl van Linné. According to this system, organisms are grouped into taxon, the orderly arrangement of organisms. The highest level of taxons will be domain or kingdom. Domain or kingdom will include all organisms in this world. The lowest level of taxons will be species. Species is only a group of one type of organism. Example, human. So human is a species. We will use human for examples of taxonomy. So classification of humans will be like this. The highest level is domain, which is human is included in domain eukarya. What is eukarya? Eukarya is a group for all organisms that have nucleus in their cell. So human has nucleus in his cells. Therefore, human is included in domain Eukarya. Domain Eukarya has several kingdoms. There are 4 kingdoms under Eukarya domain. One of them is Animalia. Okay, so human is included in Animalia kingdom. Kingdom of Animalia consists of several phylum. One of them is Chordata. Chordata is a group of organisms that have backbone. Okay, so human has backbone, therefore human is included in phylum Chordata. Phylum Chordata has several classes. One of them is Mammalia. Mammalia is a group for animal that give birth. So human give birth. Therefore, human is included in class of Mammalia. Class Mammalia has several orders. One of them is primate, the highest order. This is a group for animal like human, chimpanzee, gorilla in this order. And then order primate has several families. One of them is family of Hominoidea, which is family of human. And then the family has several genus or genera. One of them is called Homo. Homo means human. And then from this genus, the species of human that live in this world right now is only one species, which is Homo sapiens. Human sapiens. Sapiens mean wise or kind. So this is the classification of humans used as examples of taxonomy. There are 3 domains of life in this earth. Okay. The first one is called domain bacteria. This domain includes all true bacteria, bacteria that live in normal environment, bacteria that also can cause disease. They are parasites, so included in this domain. The second domain is archaea. Archaea is for odd bacteria, which is bacteria that live in extreme environmental condition. Like, for example, bacteria that live in hot spring water. So archaea, they are mostly non-parasite, so they are free-living bacteria. The third domain is Eukarya. So Eukarya is the domain for all organisms that has nucleus and organelles in their cell, including human over here. So all animal, all plants, fungi, okay, will be included in this Eukarya domain. There are 5 kingdoms of organism. The first kingdom is Kingdom Monera. This kingdom Monera is for all bacteria. So bacteria is a group of organisms that do not have nucleus. So all of them will be in the same kingdom, which is Kingdom Monera. The next kingdoms, okay, number 2, 3, 4, and 5 will be for organism that has nucleus. Okay, so there are 4 kingdoms over here for organisms that have nucleus. Okay, so the second kingdom is called the kingdom Protista. This is for primitive organism that is really difficult to tell whether this is animal or plant. Okay, because they have characteristic of animal and some characteristic of plant. Therefore, kingdom Protista is grouped into two types. The first one is called protozoa, okay, which is protista that have characteristic of animal. So it's close to the animal organism. Okay, so they able to move, for example, they're able to swim, they're able to eat, for example. So this is protozoa. The second group of protista is called algae. This group has characteristic of plants. Like for example, they are able to do photosynthesis, they have chloroplast, they have cell wall. Okay, so this is algae. Protista that very similar to plants. The third kingdom is kingdom fungi. Okay, kingdom fungi. This include mushroom, yeast, and molds. And then the fourth kingdom is kingdom plantae. So this is include all the trees, plant, bushes, grasses, and etc. And the last kingdom is the kingdom animalia. This is include all animals, whether invertebrate or vertebrate, whether they have backbone or no backbone. So included in this kingdom animalia. The next one is about the rules in naming organism. It's called the rules in nomenclature, naming organism. Each organism has two names. It's very important. So each organism has two names, first name and second name. So this system is called a binomial system. Okay, binomial system. Bi mean two, nomial mean name. So two names. The system that give two names for each organism. The first name is the genus name. It has to be capitalized. So it has to start with capital letter. So the first name start with capital letter, like our name start with capital letter, right? So this is for the first name of species, okay? And then the second name, which is species name, it start with lowercase. Okay, so the first name start with capital letter. The second name or the start with lowercase. Both name must be italicized or underlined. So you have to choose one of these, not both of them. Okay, whether italicized or underlined. Examples: human. Okay, human is a organism, is a species. So the name of the scientific name of human is Homo sapiens. Two name, right? Homo is the first name, sapien is the second name. Look at this. The first name, remember, start with capital letter. The second name start with lowercase. Both names are italicized, okay, in type in italic. Okay. Another example is the name of the skin bacteria that live on our skin, Staphylococcus aureus. Look at this. The first name start with capital letter. The second name start with lowercase. Both names are italicized. Right? So this is the rule. So when you type or write a scientific paper and you put the species name, organism name, then you have to follow this rule. Okay, you follow this binomial system rule. I think that's enough for this lecture. And this lecture also ends chapter 1. So just reminder, chapter 1 has 4 lectures. The first lecture is Introduction to Microbiology. The second lecture is Scientific Method. The third lecture is Discoveries in Microbiology. And the fourth lecture, which is this lecture, is Taxonomy and Classification. So make sure you watch all 4 lectures for chapter 1. I will see you again on the next lecture. Goodbye. Hi there. Welcome to Medical Microbiology. Today we will have a lecture on chapter 3, which is Tools of the Laboratory. So this chapter will have 4 lectures. The first lecture is Culturing Methods. The second lecture is Microscopes. The third lecture is Staining Methods. And the last lecture is Media for Culture. So we'll start with the first lecture, which is Technique in Culturing Microbes. The technique is called 5 eyes technique because it has 5 eyes in this technique. The first eye is called inoculation. Inoculation is a process of placement of samples onto media. And then after inoculation, we will put this culture inside incubator. Okay, in the incubator. The incubator is the chamber that we can control the temperatures. Okay? So that process is called incubation. So this is the second I. After 24 or 48 hours, we take the sample out from the incubator and then we observe the result, how they look like. So from these observations, you might be able to see different type of colonies with different shape and different colors. So this process is called inspection. This is the third eye. Different color, different shape of colonies is telling us that they are different species. If we want to take one species and then culture them into a pure culture, then we go to the next process is called the isolation. So isolation is the process of isolating specific bacteria based on the colony and the shape and then put it in a new culture media. And then we put it in the incubator. After 24, 48 hours, then we will have the bacteria that will have the same colonies, the same shape of colonies, the same colors of colony. Because what? Because they are pure culture, because they are the same species. And then if we want to continue to identify what species is that, then we go to the next process. It's called the identification. There are three different methods to identify the microbes. The first one using what we call the phenotypes, which is the characteristic of bacteria or organism based on their shape, for example, colors. The second method will be using DNA. This is what we call the genotype. And the last method will be using the antigens antibodies complex. This is what we call the immunology method. So we will learn the identification of microbes in chapter 17. Now we start with the 5 eyes methods again. Start with the inoculation. Inoculations of microorganisms. So this is again placement of samples into the media, into the place where these microbes will grow. There are several types of media, like for example, this media is agar media. Okay, this is agar media. So we take a sample and put that sample on this media. This is a liquid media. So we put the sample and then put it inside this media. And this media is living media, okay, which is an egg or embryo. So this is usually for viruses. So viruses need living media like bird embryo. So this process is called the inoculation, placement of samples into the growth media. The next process is incubation. Again, incubation is placement of the culture media, okay, into the chamber. So that chamber has specific temperatures, so we can control the temperature with whether a warmer or colder temperature. It is based on what type of bacteria that will be used. Okay, so if it is bacteria that usually parasite to humans, so the temperature usually control around 37 centigrade, which is around 98 Fahrenheit. Okay. And then after incubation, we take the sample out and observe the result. So we can use our own eyes, naked eyes, to observe the growth of bacteria in this media. So what we see over here is actually the colonies of bacteria. One colony consists of millions of bacteria, and they usually create different colonies for different bacteria. Okay. So there may be round colonies with yellow color, round colony with white color, tan color, and etc. And these different colonies telling us that they are actually different bacteria. If you want to see the cell, because the cell is very small, then we have to take the samples and then put it on the slide. Okay, and then after that we observe under the microscope, and then we will see the shape of bacteria. So the cell shape may be round or maybe rod. Okay, so round cell is called coccus, the rod cell is called the bacillus, or maybe there are some cell that look like a spiral. Okay, so this is cell shape. Okay, each individual cell of bacteria. The colonies again is consist of million of cell. So we cannot really see the cell shape from the colony. This is example of observation or inspection over here. So this is, you see that the yellow colony, another yellow colony, yellow colony. So the one that has the same shape and the same color is actually the same bacteria. Okay, so this is the same species. There is one that, this is white, round white, or a little bit tan, for example. So this is also telling us this the same species. But this white and yellow, they are different species. Okay, so this is the observation of colony. This is another observation of colony. Okay, with different color. This is white color, and even it doesn't have a round shape, for example. This is telling us there are two type of bacteria, okay, on this media. And this is the same thing using the slant agar media. You see this is the white one, this is bacteria, this is another one, yellow bacteria, this is another color bacteria. So this actually three different type of bacteria. So each of this is pure culture. This is one type bacteria, this is one type of bacteria, and this is one type of bacteria. So this pure culture actually taken from this mixed culture. Okay, so for example, the yellow one is actually taken from here, isolate here into this. The white one, for example, okay, take it from here and then isolate into this media, and the red one take from here, put it here. So these three test tube over here, okay, consists of each pure culture of bacteria. Okay, so that's the inspection and the isolation. This is how to isolate that bacteria. Again, on the media, maybe we will see different type of bacteria. It's called the mixed culture. And then we take just one bacteria here, maybe yellow here, and then the white one here. Then this one is become pure culture of bacteria. Again, this is continue isolation. Again, this is mixed culture. You have yellow one. We have different color over here. So these are this actually mixed culture with two different type of bacteria. So you can take this and then put it in another media. Okay, put it here, and then all of this, after incubation will produce the same type of colony. Because what? Because they are pure culture. The same thing with the other one. Okay, so let's say this one, we take it here, we put it in different media, and then incubate them. Then you will have the same colony over here. because this is also the pure culture. So this is what we call the isolation. Isolate specific bacteria to grow a pure culture. There are several techniques for isolation. The first technique is called the strict plate technique, which is we streak the media using the inoculating loops. So usually we make 4 streak on the media. So this is the first streak like this. Okay. And I use blue color first streak. Boom, 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 boom. Okay. And then the second streak will be, as I use black color, going here. Okay. And then the third streak will be, so I use this color, going here. Okay. And the last one from here to here. Okay. So basically you will have 4 regions. Okay, 4 quadrants. The first quadrant, the blue one over here, will have a lot of bacteria because this is the first streak. Still a lot of colonies. And then on the second streak, the colony will be less. Okay. As you see over here, this is the first streak. So many colony. And then become less. And then the third one become less colony. And the fourth one is become less colony. Actually, over there, the colonies will be separated. So you will be able to see this individual colony over here. So this is actually the fourth streak over here. The first one here, you don't really see the colony. So this one is really difficult to see the colony because they are still combined together. Okay, so when you do the second streak, the third streak, and finally the fourth streak, on the fourth streak, then the colony will be separated. You should be able to see the colony easily. So that's the first technique. It's called the streak plate. The second technique is called the pore plate technique, which is dilution. Okay, you should dilute the first culture, okay, and then grow it in the agar media, and then dilute it into the second, culture, and then put it in the agar media, and then dilute it again, and then put it in the media. So number 3 over here will give us the spirited colony. So it's going to be easy to see the colonies. On the first one over here, it's really difficult to see the colony because they just combine together. Okay, so this is what we call the pore plate technique. The last one is called the spread plate technique. Okay, using, put this cultures, okay, on the agar media and then spread it with the hockey stick. Spread it evenly and then put it in the incubations, and then after 24 or 48 hours, then we observe the result, and then you will see that colony. So each small dot, yellow dot over here is one colony, and the other color is also the other colony. Okay, so this is the third technique, which is called the spread plate technique to isolate bacteria. The last I will be the identification. Okay, if we really want to know what bacteria that we have, for example, if we take a sample from the patient and we want to know what bacteria that actually causing the disease, then we have to continue with identification. Again, for the identification, there are 3 methods: using phenotype, genotype, or immunological method. Again, we will study this identification method in chapter 17. Okay, so using morphology, which is phenotype. So this is phenotype method. Okay, and then this one using DNA, it's gonna be phenotype method, right? And using the antigen antibody is called the immunological method. Okay, so this one, these are the three methods that used for identifying microbes. Now, how about this biochemical? This biochemical could be used in phenotype. Okay? Also could be used in immunological methods. Okay, it depends on what type of reaction that we use, okay, for testing. So again, for the identification of microbes, there are three methods: phenotypic, genotypic, and immunological method. These methods will be studied in chapter 17. I think that's enough for the first lecture on chapter 3. We will meet again on the next lecture, which is microscope. Goodbye. Hello there. Welcome to medical microbiology. Today we will continue chapter 3, the second lecture, which is about microscopes. So this is the picture of microscope with part of microscope. So this part over here is called the head of the microscope. So this is the head. In the head, you see this part. Okay, so this is called the eyepiece. Okay, so in the eyepiece, you see the ocular lens. Okay, see the ocular lens over here. Okay, the ocular lens has power to enlarge object. So the power of the ocular lens is usually 10 times. Okay, so 10 times power. So the ability to enlarge object 10 times bigger than the original size. Okay. And then on this part, okay, you see this part. Okay, this is still part of the head. This is what we call the nose piece. Okay, so this is the nose piece. Because this nose piece can rotate, therefore it's called the resolving nose piece. In the nose piece, you will find, usually in the microscope, there are 4 objective lenses. Okay, there are 4 objective lenses. I'm going to type over here. Okay, the first one is called the scanning lens. It has 4 power. Okay. The second one, that scanning lens is the shortest one. Okay. And then the second one It's called the low power. It has 10 times power. And the third one is called the high power objective lens. It has 40 times. Okay. And then the last one, the longest one, is called the oil immersion lens, and it has 100 power. Okay, so there are 4 objective lenses usually in the nosepiece. Scanning lens 4 power, low power lens 10 power, high power lens 40 time, and then oil immersion lens 100 time. Okay, the next part, okay, over here, the whole part over here is called the arms of the microscope. Okay, and the base, okay, of the microscope is called the base, or sometimes it's called the body of microscope. Okay, another part of microscope is this flat surface. This is what we call the stage. Okay, so the flat surface over here is called the stage, and this is the place where we put the slide and the object. On that stage, you also have what we call the mechanical stage. So okay, so mechanical stage over here. So like a clip, and when we put the slide in this mechanical stage, then we can control that slide to the left, to the right, front, or back by controlling this button. Maybe you don't see that. There is a button over here. Okay, this is the button that used for control the mechanical stage. So that button is called the mechanical stage controller. Okay. And then you will also see the knob over here. So there are 2 knobs actually, bigger one, okay, and the smaller one over here. Okay, the bigger one is called the coarse focus, and the smaller one is called the fine focus. So when you turn this knob, the bigger one, for example, it will bring the stage up and down. So up and down. Right? The fine focus also will bring the stage up and down, but very smooth movement. Okay, so the fine focus also bring the stage up and down with a very smooth movement. And the last part is this part, which is the light source. Okay, so this is the light microscope using the light as a source for transmission. Okay. There is also part over here. It's very small. You see, very small knob over here. It's called the diaphragm. This diaphragm used for open and close the light source. So if you want to have a brighter light, then you can open the diaphragm. If you want to have a dim light, then we close the diaphragm. So this is the part of the light microscope. The characteristic of microscope, there are 2 of them, okay, that telling us whether the microscope is good or not. Which is the first one is the magnification, which is the ability to enlarge object. Okay, so every microscope has this ability, but there are different type of microscope. Okay, There are some that able to enlarge very, very high power. Okay, like for example, electron microscope, it can enlarge the object until 1 million times. The light microscope usually around 1,000 times, maybe 2,000 times. Okay? That's for the light microscope. The second characteristic is the resolution or the resolving power. And this is the ability to show detail. Better microscope will give you a better detail of object, more focus of the object. But cheaper microscope, for example, only give you very low detail, like a mostly blurry object. Okay, so that's the resolution. Magnification, again, this is the ability to enlarge object. Okay, so this magnification is coming from the lens that we use in the microscope. For example, in this light microscope, remember there are two lenses that I use to see the object. The first lens is over here on the eyepiece. It's called the ocular lens. Okay, you still remember the power of ocular lens is 10, mostly 10 times. Okay, and then the second lens is over here in the nose piece. There are again mostly four of them. Okay, four lenses in the nose piece. The scanning lens. Scanning lens has power of 4 times. And the second one is the low power lens. It has 10 times power. The next one is high power lens. It has 40 times power. And the last one is the oil immersion lens. It has 100 times power. Okay. So the total magnification or the total enlargement of the object will be multiplication of this power of the ocular lens with one of these objective lens. For example, if we use the scanning lens to see the object, then the total magnification will be 4 times 10. It will be 40 times. If using the scanning lens, the total magnification will be 40 times. But if we use the low power, then the total magnification will be 10 times 10, okay, which is 100 times. If we use the high power objective lens, then the total magnification will be 10 times 40, which is 400 times. Okay. If we use the oil immersions, then the total magnification will be 10 times 100, which is 1,000 times. So this is the highest total magnification that we can get from this light microscope. 1,000 times total magnification. Okay, so this is the formula for the total magnification. Also, when we see object through this microscope, the final object that we see on our eyes will be what we call the virtual image. The image is actually opposite of the real image that we put on the slide over here. So the right part of the real image will be on the left side of the virtual image. Okay, the left side will be on the right side of the virtual image. Okay? The top of the real image will be on the bottom part of the virtual image, while the bottom part of this image, real image, will be on the top part of the virtual image. So it's going to be kind of like a mirror image. Okay, so that's what we call the virtual image. The second characteristic of microscope is resolution. Now, what is resolution? The ability to show detail, right, of the object. that is enlarged. Okay, so good resolution will be look like this. Okay, so the object will be very clear. Okay, while bad resolution will be look like this. The object is blurry actually. When we see the microscope, the object is not clear, it's blurry. So this is lower resolution. Okay, the other one over here is the higher resolution. So this resolution is actually coming from two things, which is the wavelengths of the light that transmitted through the object, so the wavelength. And the second one is the numerical aperture of that light. Okay. Now the rule is the shorter the wavelength and the higher numerical aperture will give a better resolution. Okay. Now if you see this light spectrum, okay, so you see this is 400, which is short, and this is 700, is longer wavelength. So the light from here, okay, will be better for resolution. Like a blue light, for example, it will be better for better resolution. But using the red light on the other side over here, longer wavelength will give lower resolution. Okay, so usually more expensive microscope will be able to transmit more blue light compared to the cheaper microscope. This is the magnification and resolution of the light microscope. Okay, so it can enlarge small object, but it's not that small. Okay, it can, for example, we can use microscope, the light microscope, to see bacteria, for example. Okay, so this is bacteria over here. We can see that one. Okay, we can also use to see fungi like yeast. Okay, but we cannot really use this microscope for seeing some viruses, because viruses is very small. So viruses will be here somewhere. Okay, it will not be able to see using microscope, except there are some bigger virus that we still be able to see using the light microscope. But most of the viruses will not be able to be seen under the light microscope. Okay, so outside this will be using the electron microscope, because electron microscope can enlarge object into 1,000,000 times. The light microscope only about 1,000 times or 2,000 times. Now we go to the type of microscope. Okay, there are two types. The first one is called the optical or light microscope. So this type of microscope using the light that transmitted through the specimen. So using the light source. While the second one is called the electron microscope. This one using electron to transmit the energy through the specimen. Okay, just remember, electron has shorter wavelength compared to the light. Therefore, this electron microscope will give more resolution and higher magnification. There are some type or variation in optical microscope. Mostly the one that is used in common laboratory is this one, the first type over here, the bright field light microscope. And this is the type of microscope that mostly used in regular lab. So what is bright field mean? It means that the field, the environment is, or the surrounding is bright or lighter, and the object is darker. So you see the object is darker and the surrounding will be lighter. Okay. The second type of microscope is light microscope is called dark field. This one's going to be opposite. The object is actually transparent or lighter, but the field or the surrounding is darker. The next type is called the phase contrast. Okay, this will give us several type of transmissions to the object. And so you will be able to see some structure inside the cell, for example, or inside the specimen, be under the microscope. The next one is modification of light microscope using fluorescence source. Okay, so this one using usually ultraviolet source, then you will be able to see some shiny, okay, part of the object if they able to take this fluorescence light. And then the last one is called the scanning confocal light microscope. This is a very good microscope. You can see kind of 3D object using this scanning confocal microscope. Let's see what you see under the microscope using these 5 different microscope. So under the bright field, this is how the specimens look like. Okay? So the specimen or the object is darker. So this is the object, darker. Now dark field, the object is actually lighter and the surrounding is really, really dark. Okay? The phase contrast, from using this light, you will be able to see some structure inside the object. And then fluorescence, you will be able to see some shiny yellow color, okay, on the part of the object that taken this fluorescence light. And this is the scanning confocal microscope. You see this object is look like a 3D object. And so these are different type of light or optical microscope. And again, remember the bright field is actually the cheapest one, and this is the one that usually used in regular lab. The second type of microscope is the electron microscopy. And this microscope is really, really expensive because it can enlarge the object up to 1 million times. So we should be able to see very small object using this electron microscope, like viruses, for example. Even some molecules like proteins can be also seen using the electron microscope. Okay? And also the electron microscope has shorter wavelength, okay? 100,000 times shorter than the light wave. Therefore, it will give better resolution. So better magnification or higher magnification and better resolution. There are 2 types of electron microscope. The first one is called the transmission electron microscope, or TEM. This is giving us a regular 2D object, okay, regular 2 dimensions object. The second one is scanning electron microscope, or SEM, and this is the one that can give us 3D object. So the object is really cool, look like a real 3-dimension object. Now, when we look at the object, the specimen, we have to prepare that specimen. Okay, there are 2 types of preparation. The first one is called the wet mounts, okay, or hanging drops mounts. This is usually for fresh live object. Okay, we use what we call the wet mounts or hanging drop mount. This is for live, fresh object. So you should be able to see the object if it is cell or organism, bacteria, for example, they might moving around, okay, with this wet mount preparation. The second type of preparation is what we call the fixed mounts. It means that the object actually kill using maybe heat or some chemical. So the object that we see using this preparation, this prep, will be that object. So the object is already dead. It's not living object. Now the light microscope has advantage over the electron microscope because the light microscope able to see for both living object and fixed object. Okay, so that's the advantage of optical microscope. Okay. Now, how about electron microscope? Electron microscope can only be used to see the fixed or dead object. Okay, so only that object. So electron microscope cannot be used for seeing living specimen. Okay, that's the disadvantage of electron microscope. However, the advantage of electron microscope is it is able to enlarge the object into up to 1,000,000 times and also will give us better resolution compared to the optical or light microscope. I think that's enough for microscopy. We will meet again on the next lecture, which is staining technique. Goodbye now. Hello there. Welcome to medical microbiology. So today we will continue our lecture chapter 3. Today we will discuss about staining technique. Dyes are used to create contrast by imparting color. So staining is using dyes to create a contrast on the object that we see under the microscope. There are two main types of staining. The first one is the positive staining. So positive staining, this positive staining usually use basic dye. So basic dyes are cations. It means they carry positive charge. Just remember the cell is actually more negative. So cell carry negative charge. If we stain the cell with basic dyes or positive dyes, and what happens? This positive will bind to the negative charge of the cell membrane or the cell wall. It means that the stain, okay, the dyes will stain the cell. So the cell will be stained if we use the positive staining, if we use the basic dyes. So this is examples over here. Okay, so this one, okay, so this one and this one are positive staining using basic dyes. So you see the cell is actually stained with the color. The cell stained with the color. The background is actually brighter because the background is not stained. So this is the positive staining using the basic dyes. The second type of staining is called the negative staining. It is using the acetic dyes. So acetic dye is an ion, which is negative charge. And remember again, the cells, the cell membrane, the cell wall, they carry negative charge. So what does this mean? It means that this acetic dyes, which is negative, will not bind to the negative charge of the cell. It means that the cell will not be stained, but the background will be stained. Okay, so this is the negative staining using the acidic dyes. As you see over here, the one that gets stained is actually the background. So the background has stain. The background has stain. While the cell itself is clearer, is brighter because it is not stained by the negative staining, by the acidic dyes. Over here, the cells are white or brighter because what? Because they are not stained by the acidic dyes. They are not stained by the negative staining. So this is the two important informations about the two main type of staining, whether using the basic dyes or the acidic dyes. The basic dyes will stain the cell, okay, and the acidic dyes will stain the background. Okay, now there are some variations in staining. Okay. The first one is called the simple stain. So simple stain only using one dyes, whether basic dyes or negative dyes only. Okay, it's only using one type of dyes. This is simple stains. The second type is called different stain. So this one use at least two different dyes, different type of dyes. Usually the primary, okay, the first dyes will be basic dyes. Okay, so the primary stain will be basic dyes. While the counter stain, the second dyes will be acidic dyes. Example is when we perform the gram staining. So they use two type of dyes, primary stain and counter stain. The primary stain is usually basic dyes. The counter stain usually the acidic dyes. The last one, okay, type of stains is structural stains. This is a special stains to reveal the part of the cell. Okay, using special dyes to reveal the specific structure, whether inside the cell or outside the cell. For example, if inside the cell, okay, looking for some small structure inside the cell. If it is stained outside the cell, like for example, maybe staining for flagella, okay, so called the flagellar stain, or maybe stain for the capsular stain. So using specific dyes to reveal some structure of the cell. If we look at the result, maybe look like this. So this one is simple stain using only one type of dyes. In this case, using purple dyes. Okay, this is a crystal violet dyes using one only basic dyes. It will stain this cell in the same color. And then the second one, this one is different stain. This is actually gram staining. If you see the left side over here is positive. This one is positive gram, okay, gram positive. The color is purple or blue. And this one is gram-negative bacteria. The color is red or pink. Okay, so this using two dyes, positive dyes and the counter stain. Okay. And then this is example of structural staining, which is using the special dyes to refill the capsule around the cell. If you see the line over here is actually the cell. It is protected by the capsules around it. So this type of staining reveal the capsule or showing the capsules of the bacteria. Okay, I believe this is enough for a staining procedure. We will meet again on the next lecture, which is media for culturing microbes. I'll see you and goodbye. Hello there. Welcome to Medical Microbiology. Today we will finish chapter 3. So this is actually the last lecture for chapter 3. We will discuss about media for culturing microbes. What is media? Media is the nutrients that is provided to make bacteria growings in the laboratory setting. This media can be classified according to the three properties. The first one is based on physical state. Okay. Based on this physical state, there will be three types of media. The first one is liquid. So that media is liquid. The second one is semi-solid, like gelatin. And the last one, the media is solid, very firm. The next one is based on chemical composition. Okay. Based on the chemical composition, there are only two types of media, maybe synthetics or complex. Synthetic media mean all the ingredients are defined, how much the ingredient is put in that media, for example. While complex media is the media that the ingredient is not really defined. Okay, so we don't really know how much the ingredient used for this complex media. The next one is based on function. Okay, based on function. So based on function, there are several types of media. Maybe general purpose media. This media is used for growing as many bacteria as possible. Okay, so when you take sample, for example, from the bathroom, we will use the general purpose media in order to make as many bacteria grow in that media as possible. The second one is enriched media, usually using blood, because blood has a lot of chemicals or nutrients that used for bacterial growing. The next function, for example, for selection of bacteria. Okay, this media is called the selective media. And the next one is the differential media. So this media is used for distinguishing type of bacteria that grow on that media. So the bacteria will give different color on the differential media. The next one is anaerobic media, which is the media that is used for growing bacteria that is anaerobe, the bacteria that do not require oxygen. There are some media, for example, that used to know some transport or for assay, bioassay, for example, or for calculations of bacteria, to count bacteria. So this is functional type of media. Now we go back to the physical state. Okay, the first properties. Again, based on the physical state, there will be three type of media. The first one is liquid. The liquid media usually known as broth media. So if we heard about broth media, it means the media is liquid. The second type is semi-solid media, okay, between liquid and solid. And then the last one is solid media, which is firm media. In this media is also known as agar media. So if we hear agar media, it means that media is solid. The most commonly used media, which is the nutrient broth. So this is a liquid media, or nutrient agar. This is a solid media that are most commonly used in lab. Based on the chemical composition, okay, there are gonna be two types. The first one is synthetic. Again, synthetic means all the ingredients used for making media will be known. The formula will be known. And the second one is complex media or non-synthetic media. So this complex or non-synthetic media will have ingredient that will be unknown, okay, whether the concentration or maybe the type of ingredient used. Like for example, if we put blood in that media, then that media become complex because we don't really know how much ingredients, kind of amino acid, for example, or some nutrients in the blood. Okay, so the media become complex or non-synthetic. Based on function, okay, there will be several types. First, if you want to grow as many as bacteria as possible, then we use the media known as general purpose. If we put blood or serum in the media, that media become enriched. So this is enriched media. If we put ingredient that maybe select some bacteria out, okay, inhibit the growing of some bacteria, okay, that media become selective because used for selection. The next one is differential media. This is the media that is used for distinguishing type of bacteria on the culture media. So usually different type of bacteria will give different color on the top of this media. Maybe blue color, some of them may be white, yellow, etc. So we use differential media to reveal this different color of bacteria. And the last one is carbohydrate fermentation media. So this media using carbohydrate, and then some bacteria that able to use carbohydrate for fermentation will show positive result. Usually will give different color like yellow, for example. Okay, so this is media based on function. That's enough for chapter 3. So we finish chapter 3. We will meet again on the next chapter. Goodbye. Hello there. Welcome to Medical Microbiology. Today we will discuss chapter 17, specimen collection and methods in identifying unknown microbes. For the specimen collection, this is usually the procedures that I use. For example, taking specimen or sample from the mouth, take the saliva sample, sputum, blood sample, a clean cat of urine, or using the catheter. For the nasopharynx, like for example for influenza, COVID-19, usually use a sterile swab. The throat, a sample from or specimen from the throat using sterile swab. Skin surface using sterile swab. Spinal tap, uh, feces samples, and sample from vagina using the sterile swab. Now, when we have the specimens, we can do two things. First, we can perform direct testing, okay, whether microscopic, immunological testing, or genetic testing. Or we can also cultivate the specimen and put it in the culture media, let them growing, and then isolate the specific bacteria and perform the identification. Okay, so again, there are two things can be done with the specimen, whether direct testing or cultivation. There are two categories of results, maybe presumptive data, so data that can be used for diagnosing, or maybe confirmatory data to confirm the previous data, the previous result. There are three ways for identifying unknown microbes. The first one is the phenotypic method. So this using the observable characteristic that we can see with our own eyes. Like for example, the shape, the size, the color, and etc. So this is the phenotypic method. It could be microscopic. It means that we take samples and then see it under the microscope. So we look under the microscope, what is the shape of the cell, the size, the stain reaction, whether it give you blue color or red color, or maybe the structure, right, the cell structure, whether it has capsules, flagella, and etc. So this is microscopic morphology. The second phenotypic method using macroscopic morphology. It means that we observe directly on the media. Like for example, we observe the colony, okay, maybe colony shape, colony size, colony color, and etc. Okay. And then the last one is using the psychological and biochemical characteristic. Usually we will use some chemicals we mix with the sample and see what happened with that reaction. So this is also phenotypic method. So three different way of using the phenotypic methods, maybe microscopic, macroscopic, or biochemical characteristic. The second way to identifying microbe is using gene, using the DNA. This is what we call the genotypic method. So assess the genetic makeup of the microbes in the sample. Just remember, every organism, they have different genetic makeup. So by assessing their genetic makeup, then we should be able to identify that organism or that microbes. And the last way or the last method is using what we call the immunologic reaction or method. This is usually using the blood, okay, serology, using the antibody and antigen reaction. So this is immunologic method. Okay, we start with the first method, which is the phenotypic method. Again, there are several ways using this phenotypic method. Again, phenotypic method is observations of the characteristics of the unknown organism, maybe the shape, the size, the color, etc. So if you use the direct examination, for example, from specimens that we take from the patient, we can perform this direct test. Okay. For example, from the specimen, then we make or we prep the slide for microscopic assessment using the microscope, or using special or certain type of staining like gram staining, acid fast staining, or using some immunofluorescence antibody. These are several immediate direct examination of the specimen. Or maybe the specimen can be cultivated first, culture in the lab, and then after 24, 48 hours, we test the culture media. Okay, we test the culture that growing in that media. Okay, so we isolate, for example, the specific microbes and perform biochemical testing of that growing microbes on the media. We can also perform the antigen testing on that media. So these tests, okay, coming after culturing the organisms. Or there are some miscellaneous testing, like for example, for some bacteria like Staphylococcus, Salmonella, they perform what we call the phage typing to identify these two type of bacteria, or using animal inoculation, usually for leprosy, okay, or Mycobacterium leprae that causing the leprosy, and also for Treponema pallidum, which is the bacteria that can cause the syphilis. They can also perform the antimicrobial sensitivity using some chemicals that can destroy or control the growing of specific bacteria. Like for example, to identify the Pseudomonas, Streptococcus, and Clostridium. This is the common staining and also chemical tests used in phenotypic methods. So this is especially for bacteria. Okay, there are two main type of bacteria that usually can cause disease, which is coccus bacteria. So coccus bacteria are bacteria that have round type of cell. So the cell are round if we see under the microscope. So this is coccus bacteria. The second type of bacteria that usually cause disease is rod type of bacteria. Okay, it looks like a rod, stick. So for this bacteria, the first test that usually done is the gram staining to know whether the bacteria is gram positive or gram-negative. You will perform these gram stainings during the lab experiment. So make sure you follow the instructions on the lab experiment to perform gram staining to know whether the bacteria that you have is gram positive or gram negative. So it's going to be the same for coccus bacteria and rot bacteria. The first initial test will be gram staining to know whether they are gram positive or gram negative. From that process, then, okay, if it is gram positive for coccus bacteria, for example, they're going to perform the second test is called the catalyst test. This is enzyme, okay, whether they are positive or negative. If it is negative, then usually we know that bacteria is Streptococcus. Okay. If it is positive, then we have to perform another test, which is we test whether that bacteria is aerobic, strictly aerobic. Aerobic means bacteria that live, that must live with oxygen. So they will die if oxygen non-present. Yeah? If there is no oxygen, the bacteria will be dead. So this one is Micrococcus. Okay? And then if that bacteria is anaerobic, facultative anaerobic. So facultative means this bacteria able to live with or without oxygen. Right? So if this bacteria is facultative bacteria, that it could be Staphylococcus or Planococcus. Okay, so this is identification for gram-positive coccus bacteria. If it is gram-negative, okay, that can be done with oxidase test, and then with the catalase test, the same thing with this one, okay, aerobic, anaerobic, oxidase, catalase, and then finally we will know the species of that bacteria. So if you see, this method is actually using what we call the dichotomous scheme. Okay, it's a schematic. It's like a fork. So always go two results, positive or negative, positive or negative. The same thing with the rot bacteria. If it is gram positive, then there will be test for whether that bacteria have spores or not. Right? So spore former test. If they do have spore, then they are mostly Bacillus or Clostridium. Okay? If it is non-sporal, then there will be another test, especially for the TB, okay, tuberculosis bacteria. The test is called the acid-fast test. For non-TB, then the test will be non-acid-fast test. And then we're going to also see under the microscope whether they do have regular shape or non-regular shape of bacteria. Okay, so, and after that, you should be able to find or identify the bacteria that we have in the sample. The same thing if it is rot gram-negative, then there will be aerobic and oxidase test to know whether it is Pseudomonas or alkaligene. If it is facultative, remember facultative means this bacteria able to live with or without oxygen. The next test will be whether that bacteria motile or able to move or non-motile bacteria. If it's non-motile, it is going to be Shigella, Klebsiella, and etc. Okay, so this is usually the phenotypic method used for identifying bacteria. Again, you actually will do these experiments, okay, on the lab experiment testing. Okay, so during the lab, most of the activity will be identifying the bacteria that can cause disease. For the genotypic method, there are several procedures using the genetic probes. So using some small single-stranded DNA or RNA, what we call the probes, to identify some pathogen. This could be using the method what we call the fluorescent in situ hybridization or FISH, or using pulse field gel electrophoresis. So in our class, we don't really perform this activity. But if you're taking the, for example, lab, okay, on the some medical testing, you might use this test to identify some bacteria or microbes. The second procedure using polymerase chain reaction or PCR. This is kind of the most common genetic or genotype methodology that's used right now, including for testing viruses and other microbes like bacteria. For example, if you see this, this is a virus, HIV. So HIV can be identified using the PCR. Also, Lyme disease. So Lyme disease is actually bacteria. So bacteria pathogens can be also identified during using the PCR. Human papilloma viruses, HPV, can also identified using the PCR. Same thing with the tuberculosis and hepatitis. So PCR is a very sensitive procedure to identify many type of pathogenic microbes. The next one, usually using what we call the RNA sequencing. This is usually for knowing the relationship between one organism with another organism. So this is using the RNA sequencing. The last methodology used for identifying microbe is using what we call the immunologic methods. There are several procedures. The first one is called the serology. So serology is using serum. Okay, so this serum, will always done outside the patient's body. So this is what we call the in vitro. So in vitro means the tests that are done outside the patient body, and the doctor will take the serum from the patient and then perform tests on that serum. The next procedure is antigen antibody testing. This is usually also done outside the patient body, like for example, testing the blood type using the agglutination test, using the precipitation test, Western blot, usually for identifying the proteins. So this is usually done outside the patient body too, right? They perform it in the test tube, for example. And then the last one is miscellaneous serological testing. And this test sometimes is specific to identify specific pathogen. Like for example, TPI. This is special for checking or for identifying the syphilis, which is the Treponema pallidum. This is bacteria that causing the disease called the syphilis. some in vivo testing. So in vivo means testing inside the patient body by injecting some antigen into the patient body. For example, testing for allergy or TB skin test. This is actually in vivo testing, where the doctor injecting the antigens into the patient body and then see the or observe the reaction after that injection. Or using some fluorescence antibody, so the antibody tagged with some fluorescence chemicals, and then it will be shown in the results. Or using the immunoassay, okay? If it is using the radioactive isotope to label the antibodies, that process is called the radioimmunoassay or RIA. And if they use the enzyme, specific enzyme that will bind to the antibody, then that process is going to be called the enzyme link immunosorbent assay or ELISA. Okay, so these are the immunological methods for identifying bacteria. This is additional information for identifying viruses. Okay, so for the viruses, there are several ways to identify. First, observe the sign and symptom. So we can observe this outside, in the body, for example. Like, for example, cold sore, you should be able to see the sign and symptom of cold sore, especially around the lips. Okay, the sign will be, you will see the bumpy things around the lips, and the symptoms, okay, especially for the patient, they will feel itchy around this place. Okay, and we can take sample directly from that area and then perform direct microscopic examination. Usually for viruses, they're going to use the electron microscope because viruses are very, very small. Or we can also take that sample and then cultivate, okay? Growing in a spatial media, the cultivation. Just remember, for viruses, you cannot grow them on the agar media like bacteria because they need living cell, okay, in order to grow. Therefore, the cultivation of viruses usually inside the living cell, maybe like embryo, or maybe under cell culture, okay, or maybe in the animal. And so that's the cultivation for viruses. And we can also perform the serological analysis, okay, using the serum and then test with the antigen or some chemicals, and also with genetic analysis. Like we mentioned before, usually bacteria and viruses, they can use the PCR. So PCR is one of the most sensitive tests used for identifying viruses and also some bacteria. I think that's all for chapter 17. So this is just introductions of several methods that use for identifications of microbes, including bacteria and viruses. Again, in the lab experiments, you will be able to perform that activity. And then from that activity, you should be able to get more understanding about the results, about the test, and about the diagnosing the disease, okay? And also to identify the pathogens in the samples. Okay, so I hope you will enjoy the lab experiment activities. So there will be 20 lab activities Half of them will be identifying bacteria. We'll see you again on the next chapter. Goodbye now.